Vanakkam. Welcome to this course on classics in neuroscience. The last few videos we've been looking at the physiology of the action potential or the conduction of nerve impulses across the neuronal cell membrane. In this video we'll be looking at the Goldman-Hodgkin-Katz equation, famously called as the GHK equation, the model of action potential presented by Hodgkin and Huxley, the HH action potential model, and the crucial impact and legacy of the Hodgkin and Huxley model. So, the Hodgkin and Huxley model, the theory suggested that there was a flow of ions that produced the action potential. These ions were sodium and potassium. Later chloride was included in the discussion. The initial part of the action potential, I mentioned, sodium enters, and during the falling edge of the action potential, potassium exits the cell. These details I have already shared in the videos on action potential. You must be able to check those. So, there is a need to explain potential difference that you observe during an action potential, and how we can predict potential difference that is across a cell membrane that has permeability to various ions, sodium, potassium and chloride. Initially assumed that there was constant voltage gradient between the inside and the outside of the cell, and ion movement was influenced by diffusion and electric fields. Later experiments were performed by Bernard Katz that led to the development of the Goldman-Hodgkin-Katz equation. This Goldman-Hodgkin-Katz equation, the GHK equation, essentially formalizes how membrane potential arises due to permeability of the membrane to different ions. Remember, different ions have different amounts of permeability. Selective permeability is there, but also the amount, the quantity, quantum of permeability varies, and concentration gradient. So, there are two things. One is the permeability, the other is the gradient itself. So, the gradient alone is not enough, but the permeability also comes into the picture. So, this equation essentially formalizes permeability and the concentration gradient, how both of this, the interplay between the two of these two factors for various ions, for the three ions, essentially contributes to the potential difference across the excitable cell membrane. The details of this equation, the Goldman-Hodgkin-Katz equation, are presented in a different video in a different course that I taught on neuroscience of human movement. My students will share that with you. This equation came to be known as the GHK constant field equation. Essentially producing a quantitative ionic theory of membrane potentials, very critical contribution. So, what inspired the development of the HH model? So, in the 1930s, for 1 year, Hodgkin had gone to the United States, the educational tour or sort of a sabbatical, to learn about the developments in the new world, essentially where he was introduced to the CRO and other electronic amplifier and other things. In the 1940s, that is in 1948, when he took the trip to the United States, he learned from Kenneth Cole the voltage clamp method. These methodologies were critical tools, crucial tools that were absolutely important for the development of the Hodgkin-Huxley model later. So, they performed voltage clamp experiments which were key to improving our understanding of ion specific conductance changes during action potential. They first thought that there was a carrier, there was some medium or some carrier that was taking the ions from one side to the other. It took them 2 years of data analysis to disprove this idea. Then they came up with kinetic modeling of the ion flow, so chemical kinetics. So, they introduced a mathematical model that describes movement of charged particles across a cell membrane that also had a potential difference between them. So, there is a change in the conductance. So, permeability is a variant of conductance. Another way to describe permeability is conductance. If permeability is higher for an ion, conductance is also higher. They published this in 1952 in the Journal of Physiology. This model, when it was published in 1952, detailed the dynamic ionic exchanges that happen during action potential, and essentially started the foundation of the field of electrophysiology in this line of research. They won global recognition for this, because it changed fundamental understanding of neuronal cell excitability or excitable cell excitability, and it became a crucial component of electrophysiology teaching in those days. It was very quickly, this contribution was very quickly integrated into the study of electrophysiology and neuroscience. It had immediate global visibility and on research and education of neuroscience. There was then also an experimental validation with the detailed representation of sodium and potassium ion movements during action potentials, which provided a physical basis for this empirical model or empirical testing. Later, there was an extension of this work that led to the discovery of calcium channels and permeability and conductance of calcium channels and calcium based action potentials, led to the identification of many different types of potassium currents and many different types of calcium currents. So, potassium currents come in various ways, because there is a leaky current, for example, and many other forms of potassium current and many forms of calcium currents, and unveiled a whole bunch of ion channel subunits through genetic engineering and enabled precise control over ion movements. This was later development, and also essentially formed the basis for ion channel research that underpinned current understanding of ion selectivity, gating and inactivation. So, it continues to drive search for a comprehensive theory of cellular excitability. But the question is, if sodium ions are entering from outside the neuron to inside the neuron, and if potassium ions are escaping from inside the neuron to outside the neuron, and this has happened for quite some time, then it is not possible for the resting membrane potential to be at the same level, because it is the concentration gradient across the membrane that keeps the resting membrane potential at that level. A question is, how is the resting membrane potential coming back? It was later discovered that there were some specific sodium-potassium pump that pumps sodium ions from inside of the cell to outside the cell against concentration gradient, and potassium ions from outside the cell to inside the cell again against concentration gradient. That is why it is called as a pump. This is called as sodium potassium ATPase. The detailed mechanism of how this sodium potassium ATPase works, I have discussed in another video. My students will share it with you. Please do watch it. This was first described by Jens Christian Skou in 1957, essential for maintenance of constant membrane potential across the excitable cell membrane, formed a foundational role in biological membranes and how they maintain this, and it has impact beyond neurons to include other cells such as glial cells as well. Do read up about the life of Jens Christian Skou. It is very interesting. Different Nobel laureates come from different backgrounds, and so like Jens Christian Skou comes from a very rich family, and how he describes his life. So, yeah, I used to have a yacht and we used to have this, we are having fun. So, very interesting background. Compare that with the life of other Nobel laureate, for example, Bernard Katz. Different people come from different backgrounds, but all of these are great scientists. Do read up about Jens Christian Skou and Bernard Katz. Yes. These are the references that we used in the past 5-6 videos, all the critical papers and contributions. With this, we come to the end of this video. In this video, we had a discussion of the Goldman-Hodgkin-Katz constant field equation, and the HH action potential model, that is the Hodgkin-Huxley action potential model that predicted that this was happening due to ion flow, and the crucial impact and legacy of the HH model and the sodium potassium ATPase. With this we come to the end of this video, and we also come to the end of this week's discussion. Thank you very much for your attention.